So we're very excited to have Dr. Yoon-Kyoung to join us. She's currently a full professor in biomedical engineering at Ulsan National Institute of Science and Technology, and a group leader in the Center for Soft and Living Matter at the Institute for Basic Science in the Republic of Korea. She's a member of the National Academy of Engineers of Korea, associate editor of the journal Plasmonic, a fellow of the Royal Society of Chemistry, and a board of director of the Chemical and Biological Microsystems Society. Her current research topics include Plasmonic for detecting rare biomarkers, quantitative analysis of cell migration, and systems analysis of intercellular communications. Okay, with that, Dr. Cho, is all yours.
Thank you for the introduction, Cindy, and thank you for the great opportunity to share our research in this very special event. And I'm very excited to have many audience today. So my name is again Yoon Cho, and I'm going to talk about Plasmonic for precision medicine. Before I talk about, I'd like to introduce where we are. Yes, we are in Globe, so we are midnight here, and US it's 8 a.m. in Pacific time. Ulsan is actually located south from the Seoul, about 2-hour train travel. So if you have a chance to visit Korea, you may want to contact me. We are in this beautiful campus, and last week we took a picture near the pond, and these are the real heroes who are doing research that I'm gonna share today. Cherry blossom is beautiful here. In our group, we study various topics, including soft matter and nanomaterials and intercellular communications. But today, I'd like to talk about Plasmonic for liquid biopsy applications. Some of the work that I shared today has been patented and licensed to startup companies. So this is a disclosure. So centrifugal microfluidics has been advantageous for many point-of-care diagnostic applications. Over the years, including our group and our other groups, tried to use centrifugal microfluidics to integrate many biological or other applications in an integrated format. I see a lot of advantages. Like, first of all, we don't need to worry about tubing connections or bubble formations, and mixing is relatively easy. And also we can use it for the centrifuge or imaging device. And using a single mode operation, we can do fully integrate and automate many of the biological procedures, especially sample preparation for many biomedical analysis. It can be used for biomedical applications, but also depending upon your interest, food, environment, and energy applications were also possible. But today, I'd like to focus on liquid biopsy application for precision medicine. I want you to take a look at this number. In cancer drugs, for example, 75% of people do not get benefit if they have the drug developed for general people. This number was just too high for me. Like, not only the cancer, Alzheimer's, for example, 70%. How can we improve this kind of situation? Including our group, people are trying to develop liquid biopsy tools to make a group of the people so that we can provide right drug to the right people at the right timing. So I'm going to introduce our effort using Plasmonic to make contribution to this field. To make a liquid biopsy, there are circulating biomarkers that we want to detect, namely circulating tumor cells, circulating tumor DNA, or extracellular vesicles. Sounds cool, because if we can detect these disease-related markers using circulating biomarkers, we can do monitoring more frequently and a less invasive manner. However, I mean, it's always challenging. These biomarkers are rare, heterogeneous, and also fragile. On top of that, they are together with very similar high background molecules, and therefore people call it finding a needle in a haystack problem. Yes, depending upon the biomarkers, the needs are different, but for sure we need to enrich and concentrate these rare biomarkers to make a better detection. So today I'd like to talk about some examples we developed for all actually three kinds of biomarkers using Levonordisk. Let me start with the CTC enumeration from whole blood. So this is the disk. We load the whole blood in the center of the chamber here, and then put the small tabletop centrifuge we developed. And then as this spins, whole blood filtered through this chamber, and white blood cells and red blood cells flow through this waste chamber. But those cancer cells, which are bigger than the pore diameter of this filter, they remain on top of this filter. We can wash them, we can stain them, and then we can visualize them, or we can actually individually pick these individual cells and then analyze their molecular makeups. I don't want to show movie one more time. It's going just a second. Okay. You may wonder, how come the whole blood goes in and there's no clogging and washing is so clean and fast? The secret behind is that thanks to the technology we developed, we named FAST, Fluid Assisted Separation Technology. If you put the liquid and spin it without this concept, actually liquid push it to the rim due to the centrifugal force, and the particles actually sometimes clog and you don't use the whole membrane. However, if we balance the fluid resistance and keep the chamber wet during the process of all centrifugal time, we don't need to overcome centrifugal force, and therefore the blood separation or particle separation happens at low frequency, and we can utilize the whole membrane for the filtration. So using this concept, we developed the Levonordisk that I showed before, and of course the recovery rate was really good, and we can purify the EVs and then mount it on the slide glass and then visualize them, or individually pick up those CTCs. And it's label-free, it's size-based separation. And compared to the gold standard cell search system, we could, of course, collect the many CTCs, but more importantly, I'd like to emphasize this graph here, that when we make two different bottles, it's really reproducible. And thereby, we could install this device in the hospital. And my collaborators did experiment, like the example shown here. So these patients were undergoing these different chemotherapies, and they measured number of CTCs. And one of the observations we did was that not just the number of CTCs, but their change was actually more correlated with the disease progress. And also, not only the single CTCs, but the CTC clusters we observed a lot. And there are many cases together with the platelets. And those patients who has a platelet-associated CTC clusters showed a poor outcome. But CTCs are really rare. More common marker for liquid biopsy is the cell-free DNA. And even though there are like a really nice automated equipment for the cell-free DNA isolation, we thought if we can have this point-of-care DNA isolation device, instead of shipping the whole blood, we can ship the purified and enriched the DNA for the further analysis. So here we developed a Levonordisk to separate ctDNA from the whole blood. The plasma separation happens, and these black dots are valve, and plasma go to next chamber with it's preloaded with lysis buffer and binding buffer and so on. They go to the glass bead, and the DNA or the glass is bound. And this whole process takes like 30 minutes without hand touch, fully automated. Using this device, we measured a lot of ctDNA from the non-small cell lung cancer patient. And this is a really nice example to highlight the importance of the precision medicine. So in this case, this patient were like diagnosed as a non-small cell lung cancer with the EGFR mutation. And got the chemotherapy. It was lucky in the beginning, tumor size got reduced, and also the mutation frequency of the L85R was decreased. However, sooner or later, as you can see, the tumor size got bigger and also mutation frequency got bigger. More importantly, from this ctDNA, we could see the emergence of the T790M mutation. Several months later, doctor did a second tissue biopsy and confirmed that the T790M mutation, and she changed the drug to the third generation drug, and now the patient go back to the stable condition again. So one can think about, like, what if we can detect this mutation much earlier and do the things earlier without waiting this several months. So that could like really motivate my student to do further research. DNA is good, but DNA is only the DNA. And more recently, many people are interested in extracellular vesicles, and we use the similar concept to isolate these tiny nano-sized vesicles who actually have a lot of traits of the cells, of the original cells, and also not only the DNA, it could have a microRNA and messenger RNA and proteins. So we developed the level disk called the ExoDisk to isolate EVs from samples like urine, bronchial washing, or blood, etc. So let me share the story with the urine sample first. So here the disk designed with the sample loaded, and then two filters are sequentially located. And so flow coming through the first filter with a pore diameter of 600 nanometer, and then second filter with the 20 nanometer. So if you filter them, those particles size bigger than 20 nanometer and smaller than 600 nanometer, they are enriched on top of this second filter. We further utilize this filter one more time. So those filters are actually captured on top of this filter. So after washing and so on, we can use it for the ELISA. So particles EVs are captured and then wash it, and then we introduce secondary antibody and substrate reactions, and we can see those color changes, just like the normal ELISA, but in this case, there is no capture, and it's in a solution. So if you see the movie, urine, like the cells are sedimented, the clear urine go to two filters sequentially, the particles are captured and washed twice, and then those are enriched in this collection chamber. You notice that starting from the yellow solution and going to the pure, like nice blue solution here. Okay. So now I'm going to show you another precision medicine example with a prostate cancer case. It's the second highest cancer mortality in U.S. men. It's like in the lung cancer sample, I showed the importance of choosing the right drug. Here, another importance of example showing that maybe you have right to not to choose the drug. So in this case, the prostate cancer patient, if they the cancer progress, the patient can have actual benefit from the anti-androgen therapy. But if the patient have this androgen receptor variation, they do not get benefit from this anti-androgen therapy. This is FDA approved if the ARV7 is detected from the either tissue or CTCs. Here we hypothesize that what if we can measure this ARV7 from the urinary EVs. So we took a urine, filtered the EVs using ExoDisk, and then using the ddPCR, we tried to detect ARV7. Yes, when we tested healthy donors' urine and hormone-sensitive prostate cancer's urine and CRPC patient urine, as you can see in the orange chart here, the ARV7 RNA occurrence was increased. Not only that, the wild type was reduced. So we could see not only for the drug choice, but also maybe we can see the progress of disease by measuring ARV7 RNA from urinary EVs. One of collaborator in the hospital suggests that what if we can use bronchial washing fluid? Maybe we can have a better sensitivity in those mutation detection. So here we took bronchial washing fluid from the lung cancer patient, and then using ExoDisk again, on top of the filter, we can collect the EVs, and then from the bottom of the filter, we can get EV excluded the DNA portion, and then we tested EGFR mutation for the non-small cell lung cancer patient. To make a long story short, yes, bronchial washing-derived EV showed much higher detection sensitivity compared to the plasma DNA or tissue. But also, very importantly, when we tested these 3 different samples all together, sometimes, yes, the 85R was 19 deletion mutations were detected from all 3 different samples, but T790M was detected only from the bronchial washing-derived EVs, not from the plasma ctDNA or tissue. So first we were wondering whether it was false positive, but later, several months later, it was confirmed in either plasma or tissue samples, which may suggest that the disease-related mutations coming, we could, from the bronchial washing sample, we could detect these mutations much earlier. We also developed the level of this that we can isolate EVs from the blood plasma. So in this case, from teeny tiny, like 100 microliter of blood from the mouse, which we inoculated tumor. And as you can see here, either like the tumor volume, as the tumor volume increases, the CD9 or CD81, the normal EV markers didn't change much, but tumor-associated markers was increased as early as from ever like 1 or 2 weeks after the tumor inoculation, which again highlight that maybe those EVs can be a nice biomarker to trace the disease. We were actually questioning whether, like, is it really necessary to isolate EVs? Here we developed the electrochemical sensor where we modified the surface to make it really nanoporous and nanostructured, so that we could use undiluted plasma directly and capture the EVs and detect the EVs. In this picture on the right corner, you can see that when we use the plasma directly or PSA, in the normal gold electrode, resistance is really increased high, but nanostructured and nanogold, the resistance change was not really high, so we could use it for the electrochemical detection of those protein EV markers. Now, when, like prostate cancer, when we have well-known tumor-related biomarkers, it's relatively easy. However, most of the cancer cells, we really do not know which EV is actually coming from the tumor or normal cells. So inspired by the observation that we learned from the CTC studies, you know, those CTCs were actually traveling together with the platelets. So we prepared the platelets and then coated on the glass surface to make a platelet membrane surfaces, and then hypothesized that whether those tumor, whether they are coming from cancer site or normal cells, whether there is adhesion differences. So we made a platelet membrane chip and then treat the EVs, and then introduce these gold nanoparticles or gold nanorod and did a dark field imaging. And when we treated the EVs from the normal cells, we didn't see much binding event. However, when we treated cancer-derived EVs, yes, we could see countless many EVs. And as we increase the concentration, yes, the binding event increases. And when we compare to the normal cell-driven EVs, cancer cell-driven EVs or metastatic cancer cell-driven EVs, we could see the more binding event. And it was further confirmed when we tested with the prostate cancer patient plasma samples. So maybe this tumor and platelet interaction could be used for cancer detection, and we are doing further study. We thought we asked another question in this case. You know, EVs may have doing some role in the organotropic cancer metastasis. There were previous studies saying that depending upon the type of EVs, they contribute for the liver or lung cancer metastasis. So here, to test that hypothesis, we prepared organ on a chip, and in fact 3D liver on a chip. And in this structure membrane, and then on top of the membrane, we sort of mimic the hepatic cynoid, have this synoidal, liver synoidal endothelial cells. And then underneath of the membrane, we mimic the specificity of the hepatocyte. And so preparing this 3D structure of the human liver model, and then flow EVs first. And then depending the type of EVs, we tested whether the adhesion of tumor cell is influenced or not. Yes, the hypothesis was right, and the number of adhesion was actually more enhanced when we had a 3D liver chip model compared to just the one type of liver cells. And when we tested different cell types, like for example, breast cancer cells or pancreatic cancer cells, which shows liver metastasis is open, we could see more enhanced binding of the cancer cells when they are pre-treated with this pancreatic cancer cell-driven EVs. On the other hand, when we treated brain-derived EVs, which is not common to liver metastasis, we didn't see much changes. When we further tested with the patient sample, breast cancer sample with liver matter or non-matter, we could see the differences again. But we need to do more study because this number of cohort is relatively small, but it suggests maybe we can have a tool to predict or measure metastasis. That was the impact of this study. So far, I introduced a lot of centrifugal devices for the diagnostics, etc. But unfortunately, all of them, I need to have electricity. In low resource limited settings, I cannot use none of these. One day, I got really inspired by this beautiful hand power centrifuge developed by Professor Manu Prakasi at Stanford. It can spin like up to like really high spin speed, and we can separate blood plasma, etc. So we thought about it, and we come up with the idea that maybe we can detect UTI. And UTI is a common disease, but because antibiotics are usually used, and so it actually contributes for the development of the resistance. So in India, for example, if the patient has a symptom of UTI, they go to hospital and then just get the antibiotics. And then they send the sample to the clinical labs, etc. And then several days later, you get the result and find out that, oh no, I didn't need to take antibiotics, right? We could reduce this kind of overusage of antibiotics. That was the motivation of the study. And this is the fidget spinner, maybe, like the toy for the children. We kind of modified this fidget spinner and make a diagnostic fidget spinner. And with just less than $1, we can detect UTI and also do the very rapid antibiotic susceptibility test. So you may wonder, like some people can spin really quick and carefully and some are not. How can I enrich the bacteria with, just a hand tweak? Yes, because of those fast technology that I introduced for the CTC application, we need just 3 spins. No matter whether you are careful or lousy, we could isolate enriched bacteria from 1 ml of urine. And after this filtration, we introduced the WST dye. Then when only the bacteria is live, it shows orange color, and with the sensitivity less than 1,000 CFU per ml. And if we do RPA and lateral flow assay, we can go down to 10 CFU per ml. It is only sensitive to bacteria, but not sensitive to the mammalian cell. So using this simple device, like a fidget spinner and urine sample and dye, my student went to India and tested like 39 patients in a week. And yes, after the test, 21 people actually didn't need to take antibiotics. We could contribute for the stopping overuse of antibiotics. And we thought one step further, like once the bacteria is confirmed, we can treat the antibiotics and spin one more time. If the bacteria is susceptible to the antibiotics, the color is reduced. However, if they are resistant, color remains. So we took many more clinical isolate samples and then confirmed that 100% sensitivity and specificity. And again, this AST test without electricity, within 2 hours, we could do that, while conventional method takes several days. And this was a nice example to show that WHO assured technology, which might be have an impact in really low resource limited settings. So with that, I hope I convinced you that Lebanese can be a platform technology to be used for many different biomarker detection. And sample preparation can be easily integrated on a disk so that it can be used for the point of care diagnostics. So with that, I'd like to thank you, my current and former student. And we are in Korea, but in a very international environment, and we are looking for always postdoctoral students, all kinds of levels. So thank you for your attention, and I'll be happy to answer 1 or 2 questions if you may have. Thank you.
Great. Thank you so much for the wonderful talk.